All right, we're back again. We ended right here with the codons that you have in messenger RNA. We ended saying that you had 20 naturally occurring amino acids, but you had 64 possible codons in messenger RNA, which would have also been 64 in the DNA triplet code. We said that, therefore, if you have 64 to code for 20, that you have several coding for the same amino acid. But can any of those codes code for another one? No. So therefore, the statement in most books is you have redundancy, but not ambiguity. It's not ambiguous, but it is redundant. And this shows the various ones. Okay? All right. So now we go to what I wrote out to look at the steps of protein synthesis. The steps of protein synthesis. So that's where we are now. Okay. In the steps of protein synthesis, step 1, you have to find the proper gene for the proper polypeptide among the 23 pairs of genetic material. This is occurring in the cell nucleus. So the first thing that has to happen is, where is the gene? Where is the gene? Now you have 23 file drawers, let's say. 23 file drawers. And each file drawer is duplicate. One came from mom, one came from dad. But 23 file drawers, they're duplicate. You're trying to find the proper gene. So the cell has to look into which of the 23 file drawers is the gene. Okay? Now, the 23 file cabinets. All right. Now, once it finds, let's say it's chromosome pair 2, then it has to decide, am I going to read mom's side or dad's side? Now, that's okay if both of them are fine, but sometimes one is dominant and the other is recessive. Hear my word. Sometimes one is dominant. Maybe mom gave you dominant for this particular characteristic, and dad gave you recessive. So let's say the protein. I'm just going to pick a protein. Let's say insulin. So you would be looking for the blueprint of insulin. So you'd go through the 23 pairs of genetic material, find whatever chromosome contains the genes to make insulin. But you would definitely at least have 2. You'd have one from mom and one from dad. If both of them are fine, you could pick either one, the cell could. If one's dominant, the other's recessive, we would try to pick the dominant one. So now I'm at the cabinet. Now I'm at the file cabinet. But now I got to look for the file drawer. So now within the cabinet, we're now searching through the genetic material to find the proper gene locus. We're searching through to find the proper gene locus within the DNA. The proper gene locus within the DNA. So that's our next job of the 23, is to look for where the gene is on the genetic material. Where's the gene? Because when you look at it, it's looking at where in the genetic, let's say it was chromosome pair 2, where is the, what we call gene loci, the gene loci, okay? Now, once you find that, then what's going to happen is, I'm going to come back here, once you find the proper gene loci, then remember, you're reading from a 3 to 5, but the DNA is 3 to 5 on one side, 5 to 3 on the other. So what will happen now is you're looking to see which of the RNAs are the correct, which of the DNA strand is the correct. Okay. Because on one strand, you could have AACAT. On the other, you'd have the opposite because they're bonded. So you have to look at which side of the DNA, the cell wood, do you read from. Because if you're reading in a 3 to 5 direction, one side is inverse of the other side. So my point is, you technically have two genes even in that same region of DNA. Now, so we're looking for insulin. Let's say this strand here, once you split it up, this strand here has the gene for insulin. This may be another gene. So in that particular case, this strand here would be the sense strand and this would be the non-sense. Only because this is the side we're looking for. So let me go back again. You go through the 23 pairs of chromosomes. You then find the one that has the gene to make insulin. You then find the location in the DNA, what we call the gene loci, where it is. But when you find that, it's double-stranded. One is going 5 to 3 in one direction, one is going 5 to 3 in the opposite. The reader is going to read from 3 to 5. So technically, you have two genes even in that same region. You possibly could. So you're trying to find which side of the ladder is the gene you're looking for. Which side of the ladder? The side of the ladder involved in the gene you're looking for currently is called the sense strand. The side that's not is called the non-sense. It might be vice versa. If you're going for glucagon, which is another protein, maybe the other side have it. And then that side would be called the sense strand, and the other would be called the non-sense strand. Okay? So it's a big feature here just to find where the gene is within there. Okay. Now, number 2. You now want to read the gene's reading frame using the enzyme RNA polymerase, because you're going to make an RNA copy to the DNA. You're going to make an RNA copy to the DNA. The copy you're making is called messenger RNA. The copy you're making is called messenger RNA. The messenger RNA has the codons. So you made, and it's the inverse. So if this is, let's say, the strand of DNA, here would be the RNA you're making. If this had a T, you'd put an A. If this had an A, you'd put a U. Remember our bonding pattern. Go back and look at that. So you're making an inverse copy. The messenger RNA is making a copy, but it's making an inverse. Then it will be flipped later in the cytoplasm. So read the gene's DNA using the enzyme RNA, thus making an RNA copy of DNA. Since the action is one nucleotide being copied to another, that particular process is called transcription. Transcription is where you're transcribing one language to the same language. One language, so read this in the book, to the same language. Transcription. So the making of messenger RNA off of DNA is called transcription. Okay? And you're making the messenger RNA, so you're making a copy. Okay? Now, the copy that you're making has too many nucleotides. So let's say insulin, which has far more, had 100 amino acids. It has far more than that. When the messenger RNA is made in that gene, it's going to read 300 or 400. Now that's too many. So you're making what we call a pre-messenger RNA. You're going to have to cut out some areas within that. You're going to have to cut some areas out within that. Right now, the message, if you took it in the cytoplasm and read from this long message, you would make an ineffective insulin. So what you're doing is you got extra regions in there when you read on it. Those extra regions are called introns. The regions that you want to read are called exons, E-X-O-N-S. Okay? So introns and exons. What's going to happen in the nucleus is you're going to have to cut out the parts you don't want, known as splicing, and hook the ones that you do want together. Cut out the ones you don't, that is not needed, and hook the others together. That's what we call splicing. Splicing. That is occurring, let's go here. Here's the splicing process. See, this is a pre-messenger RNA. You have exons, intron, exon. What I need to do while yet in the nucleus is cut the introns out and hook the exons together. That's called splicing. A complex of enzymes in the nucleus is going to do that, called a spliceosome. You do not want the message to exit the nucleus. Let me find a picture. You do not want the message to exit the nucleus into the cytoplasm until it's undergone RNA processing. That means take the pre-messenger RNA, cut out the introns, hook together the exons, and now you got a good message. And then the messenger RNA can exit through the nuclear pores. So again, transcription is the job of reading from DNA nucleotide talk into RNA nucleotide talk. But when you read into the RNA nucleotide talk, you have some extra areas in there that must be cut out. The extra areas are called introns. The spliceosome I showed you has the job of going in and removing the introns and hooking together the exons, which is a good message. Then it will exit. Now, there still will be some extra pieces at the end that we call the capping units, but at least the message on the interior is good and clean. But the capping units will still be at the end here. Okay, so we'll go back over here. So we have the RNA modifications. The newly formed RNA is termed pre-RNA. In it, it must be modified in two ways. Certain regions in the RNA must be cut off, and some capping nucleotides must be enzymatically attached to the end of that. So the capping right there would occur. Now, the capping is like a book cover, because when you take it in here, it's going to be hostile, and you don't want to fray off the book cover and start destroying some of the message, we'll say. The other thing the capping units are going to do is help draw the message to the ribosome. And what it's going to do, the message will sit on the small subunit. So let's go further. So splicing. The newly formed messenger RNA has some intentionally added nucleotides over and above those needed. The nucleotides are in the middle of the message. These extra nucleotides are called introns, known as intervening regions, contracted terms. The nucleotides are called exons, the ones you want. So you want to cut out the introns and hook together the exons to give a nice message. And that message would then be good messenger RNA. So then the only thing you would have are the capping units, which were enzymatically placed. Okay, we'll go here. So each end of the pre-RNA is modified in a particular way. The 5' end, see, when you look at the message, one end is 5', because but remember the 3 to 5 hookup? 3 to 5 hookup of the phosphate to the sugar. One is the 5', the other is the 3'. You don't have to get heavy in this, but you have a capping unit on the end. That's all you really need to know. Once the modifications of the messenger RNA are completed, the messenger RNA can exit the nucleus and enter the cytoplasm. It will exit the nucleus through the nuclear pores. That's one of the jobs of nuclear pores. The other job, remember, was to export developed ribosomes, but they would export in small and large subunits. Then once you get here in the cytoplasm, some proteins called chaperone proteins will help take the messenger RNA to the small subunit of the ribosome. So it's going to initially attach on the small subunit of the ribosome. The small subunit of the ribosome acts as a construction table for the newly forming polypeptide to be made. Okay? So we're going to end right here at that step there. We're going to end right there at that step there and pick it up with the next one. We may have to have 4 of these, 4 videos.